All right. Let's start to summarize some of these, challenging examples, etc. I probably should have mentioned this before, but the other night is from the Norwegian Sequencing Center and at the University of Oslo. And we are together with a node at the hospital that runs most of the sequencing. But we are the largest sequencing core in Norway, so we have a lot of different projects. The Research Council of Norway is, of course, the main contributor, but also the health authority. We are currently involved in the ERGA and the Earth Biogenome and the BGE. Yeah, so we kind of got funded for this Earth Biogenome project in Norway. We always have to have mood on everything we do. It was kind of a 3 million euros initial funding to do roughly 150 species. Yeah, we got the other universities on board, as well as, some more everything from consulting to fisheries and computing interest together with us. It hasn't really sparked that, huge interest within, commercial companies. They are clearly looking for what's in it for us type of thing, which is kind of understandable, I think. Okay, so the challenge examples for HiC. Once again, is it something you are doing in the lab? Or is it something with the samples that have been sent to you, or a combination? You never know. But you have to be honest about it. That's for sure. So how do we describe a challenge sample? I mean, it depends on who you ask, right? Is it an experienced user that have handled these samples quite many times? They're not challenging anymore. So it depends on the level of experience within the lab and maybe also with the analysis. Is it the core facility, or is it the user that determines if this is a good sample or not, or challenging? It's difficult to say. What we have realized is that small species, meaning low input, which is the same as low number of cells, is challenging, usually. It's not the same as low quality. Very often you will see, whether it's HiC or sequencing, is that it's a bit dodgy quality. But let's call it a low quality sample or challenging sample and then go on. That you have to avoid. Because even you see it even on the ultra low input things for long read sequencing, for instance, PacBio. The input is low, but you really have to have long fragments to be able to perform a good library. So it's very easy to start thinking about lowering the kind of the quality thinking when you have something that is also low input. Or is it just little material available? Have they sent you a tiny skin sample from a large mammal? Why should you work with that? We see that as well, right? Well, then you should probably considering the sampling you're doing. Just by looking at the sample, you can identify if this is or not. How well is it actually preserved? From other difficult samples, or by just looking at it, that the texture or the composition might indicate something. I mean, a sponge, for instance, a snail, an algae. It's kind of easy to to realize that this might be somewhat challenging. So the commonality is that during the prepping, they will give you some sort of challenge. And then you have these more unknown things that you don't really know from the start. And that's which kind of reagents, chemicals, biochemistry does this sample have? Inhibitors, for instance, that can mess up your ligation reactions. Or, like we heard with our talk, nucleases. As soon as you start growing something, especially if it's very few cells, you're dramatically reducing the numbers of available nuclei to cross-link and go on with. So as we already heard, the preservation is of course central. Also, how do you transport sample from the field to the lab or even to the core? I mean, if you want to do a high C, you don't take a small ice cube and send it away. Some do. And also the long-term storage, always aim at these -80, -70 freezers and not do any freeze-thaw cycles. We have experienced that samples that shouldn't have been thawed, they seem to be anyway when you start digging into it. It happens. The prepping, as Arjen said, this thawing could be problematic. We have seen it also in mammal blood, dog blood. I think also Korean has some issues with dog blood. Blood that has been put in EDTA tubes, which is the way most human blood also is treated today. It's not the best solution for long-term storage. Why? Because it doesn't inhibit all nucleus activity, especially, and one thaw cycle might be enough, and then you have drastically reduced the quality of the DNA, for instance. And while you start thawing it, the nucleus activity starts again. So they have put in kind of a nuclease inhibitor into the frozen sample, and then you get a much better result. The integrity of the DNA is a lot better. And this is on so-called EDTA blood. So it's something to pay attention to. You also have to pay attention to what kind of nucleus inhibitors are you then using, because like in the paper you wrote with
DNA guard, it skews results towards increased trans interactions. So the trans interactions that you see are not real.
No.
So we have a paper about that. I'll link to it.
So
for the vertebrate samples. So RNA later and DNA guard, they are doing something.
Yeah.
That makes sample unusable for high C.
Yeah. So we also need now, due to this course, that the importance of having proper chemicals while you do the prep, the formalin, for instance, proper grinding, and all that. And then this constant sample quality evaluation, where you have to be honest with what you're doing, it's really important. And I believe that you need to have a kind of a more team-like structure to have feedback from lab to the bioinformatician and vice versa. I think that's when you kind of are able to make those small adjustments that can actually put this forward. So this is kind of a summary of that, how to minimize some of the challenges. And just simply have to say, no, we are not doing this one, if you are in doubt. You will meet people that are not willing to accept that. And that's this kind of academic pride people that will also try to learn you how to do DNA isolation and qPCRs, even though you have been delivering Illumina libraries with accurate concentration for 10 years. They know better for some reason. So at least if you're running a core, you have to have these minimum guidelines and stick to them. And be also very informative towards new users and spend the time that they actually get it. And be friendly. And then people are willing to follow these standards, usually. And then bug your bioinformatics team to make sure that you get proper QCing all the things you're doing, because then you can also start thinking, how should I improve this in the lab? Then you have, of course, the option of checking with others and on real manuscripts and all that, papers, and don't be afraid to ask others. And finally, the things that we have touched upon, the DNA quality. How was the DNA isolation done? Did that show any kind of trouble? It will usually inform you about if it's necessary to take extra step for the HiC. It doesn't have to be the case. Sometimes sequencing in just long-read sequencing is more difficult than the HiC, but anyway, it's a good thing that you kind of have it in mind at least. If this was a problematic species for sequencing, then think twice before you just rush on with the HiC. Like these samples I put there. This is springtails, columbola. This is the DNA. Blue or black. The whole palette in that color. This is the DNA. The library illusion, final libraries look almost the same. One of them did not make it through sequencing. Guess what?
Yeah, black.
It's actually the black one, but the other ones worked. So you never know, right? But usually there are some indication at least. So pay attention to these kind of phenotypes of your DNA or your material. All right.